Hello, and welcome to another video of mine. In this video, I want to talk a little bit about cell division, how the cell undergoes division or divides. And specifically, want to start with mitosis, what we call mitosis, or the somatic cell division. So let's look at mitosis. I want to look at mitosis. Okay. So as I said, another term for mitosis is somatic cell division. So also known as somatic cell division, right? So somatic cell division means the division that goes within the body cells. Okay, anytime you see somatic, this refers to like a body cells. Okay. Great. So what is mitosis all about? Basically, mitosis is involved in the growth of tissues, right? When the body is growing, the cells have to divide. So that process is what we refer to as mitosis. So it's more for growth. Of cells. There's another one called meiosis, but we'll talk about that in another lesson. There are basically 4 stages of mitosis. So the mitosis, the stages of mitosis. 1, we have what we call the prophase. 2, we have metaphase. 3, you have Anaphase. And then 4, you have Telophase. So these are the 4 stages of mitosis, or what some people call the phases of mitosis. Okay, so let's narrow down to what happens in each stage. So let's look at prophase. So let's begin with a cell that looks like this. And the cell, as we know, has a nucleus in it. And let's assume that we have 2 sets of chromosomes here. So this is a chromosome. I'm going to use another color for the other chromosome. So let's say that is red. Okay. And one more, another black one. Okay, good. So this is a nucleus. Let's put nucleolus here. nucleolus. Okay. The nucleolus is involved in cell division. And then over here, I'll put the centrioles. Also involved in cell division. Centrioles. So during prophase, what happens is that each chromosome makes an exact duplicate of itself, right? So prior to mitosis, each chromosome makes a copy of itself. Okay. So each one will duplicate itself. So this is going to duplicate itself. That will duplicate itself. This will duplicate itself. And this will duplicate itself. Okay. And the chromosomes thicken. And then they coil. Chromosomes thicken, become thicker, and coil. Then in the early prophase, the centrioles divide. So the centrioles will divide. Let's show that here. Centrioles divide. To form what we call the asters. So the asters are like the spindles that will be formed. And then they move apart. Now the nuclear membrane here breaks down, it disintegrates, right? So you're gonna have disintegration of the nuclear membrane. So let's put this here as a nuclear membrane. So that breaks down during the early prophase. So we're gonna have these chromosomes. Like that. Okay, so let's put down some few notes here. In early prophase, the centrioles which have divided form asters and move apart. Okay, so that's the first thing we said. Then secondly, the nuclear membrane begins to disintegrate. Okay. So that's what happens in prophase, right? The early part of prophase. Then in the late part of prophase, so let's look at the late prophase. So we can put here, let's write that early. Early part. And then this will be late prophase. Late prophase. What happens here? So in late prophase, the centrioles and asters are at the opposite poles, right? So they be at the opposite poles. So I have in the diagram form, let's put this down here. So what we're saying is that these centrioles will be at the opposite poles. Remember, they move apart during the early phase. So they move to opposite poles like this. Okay. Then the nucleolus and the nuclear membrane, they have almost completely disappeared by this state. Okay, so we're going to have our chromosomes. Okay, so by that time, the membrane is almost gone. Nuclear membrane is almost gone. Okay, so let's write these notes here and say that in late prophase, the central and asters are at opposite poles. The nucleolus and nuclear membrane have almost completely disappeared. Okay. So now you see the spindles will be forming, getting longer here. The spindles. They will get attached to the chromosomes shortly. Then we enter the phase called metaphase, the second phase of mitosis. So metaphase, let's look at what happens as metaphase. So during metaphase, the double chromosomes their centromeres attached to the spindle fibers, and then they line up at the mid point of the cell, the middle of the cell, which we call the equator of the cell, right? They all line up, have a single row. So you have something that looks like this. Then the chromosomes all line up at the equator. Okay, they have your spindles attached to what you call the centromeres. Attached. Okay. So let's write some few notes down. One, the doubled chromosomes have spindles attached to the centromere. All right, so remember the centromeres are where the chromosomes are, the chromatids, what we call the daughter chromatids. They are attached to each other at the centromeres, right? And then we said that they line up at the equator. The chromosomes line up at the equator of the cell. So you have a single file like this. Now we're going to enter into another phase, which is going to be the anaphase. The anaphase. Okay. So this has the early parts and the late parts. So let's look at the early parts. Early anaphase. Now, during early anaphase, you have the chromosomes splitting up. So the spindles will shorten, and the spindle shortening and pulling the chromosomes apart like that. Let's use the black one for this. The chromosomes are moving apart. The spindles are shortening and pulling them to opposite poles. Okay. So let's put these notes here and say that in early anaphase, the centromeres split. Then you have half the chromosomes move to one pole and then half to the other pole. as you can see here. So they are being drawn to the opposite poles here. So that's what happens during early Anaphase. So let's move on to the late Anaphase. Late Anaphase. So during late Anaphase, what's happening? You have the chromosomes have almost reached their respective poles. All right. So you have something that looks like this. And then you see that the cell membrane begins to pinch at the center. So let's see, you have this. Something that looks like this. So they're almost at the poles. Okay. And then it begins to pinch. Membrane pinches here. Okay. So let's put down and say that in late Anaphase, the chromosomes have almost reached their respective poles. Then the cell membrane begins to pinch at the center. Then finally, we enter into the telophase, which is the last phase of mitosis. Okay. So during telophase, we're going to end up with a diagram that looks like this. To have this, the other one, then this one, and that one. And over here, we have the same thing. Okay. So the cell membrane completely or completes its constriction in telophase. So let's put down that the cell membrane completes constriction at telophase. Okay. Next, you have the nuclear membrane. Form around these separated chromosomes. Okay. So that's what happens during telophase. So at the end of mitosis, there are two cells in the same structures, with the same structures and the number of chromosomes as the parents. Good. So we end up with a cell that looks like this. Nuclear membrane formed. The chromosomes in there. So end up the same number of chromosomes. All right. So if we start with the haploid number of chromosomes, sorry, we start with a diploid number of chromosomes. Which in human beings, you have 23 pairs. Or 46. Chromosomes. At the end of mitosis, you end up with the same diploid number. The same 23 pairs. Or 46 chromosomes. Okay, very important to note this. The number of chromosomes hasn't changed based on what we just done, right? So that's how mitosis occurs in our body. Hope you enjoyed this video.